Hello friends, welcome back to another video tutorial from Arun's Biology. In today's lecture, we'll discuss about the tryptophan operon. So let's begin the topic. So tryptophan operon is basically a operational unit that is concerned with an anabolic or biosynthetic pathway, and it helps in the biosynthesis of an amino acid, that is tryptophan. Thus the name of this operon is tryptophan operon. Now, tryptophan operon is basically regarded as a repressible operon. That means the presence of a repressor molecule will inhibit the transcription of the structural genes present in this operon. Okay, as a result, the formation of tryptophan or its biosynthesis will be inhibited. That's why it is called repressible. Now it is under positive regulation. That means under basal condition or under normal situation, when tryptophan is not available to the cell, this operon system becomes switched on and it helps in the biosynthesis of tryptophan. Now when tryptophan becomes available to the cell or the cell itself synthesizes tryptophan, then this operon undergoes switch off condition as the tryptophan biosynthesis is not required. Thus it is called positive regulation. The overall knowledge about the tryptophan operon comes from the discovery of the Charles Janowski and his co-workers. Now just have a look on the different components of this tryptophan operon. So what are the components? So components are the repressor sequence, the operator sequence, the leader sequence, promoter sequence, and the structural genes. Now they are arranged in this manner. So this is the repressor sequence, this is the promoter and operator. Now this promoter and operator, these two sequences are actually called the regulatory sequences because they help in regulation of tryptophan operon. This is the promoter sequence that will bind to the RNA polymerase and that stimulate the transcription of these structural genes. Now this is the leader sequence of tryptophan operon, which is having immense importance in the regulation of tryptophan operon through attenuation. These are the 5 structural genes starting from tryptophan E to tryptophan A, and these 5 structural genes help in the synthesis of 5 different enzymes. Now what are these enzymes? Tryptophan E helps in the synthesis of the anthranilate synthase component 1. Tryptophan D synthesizes anthranilate synthase component 2, or it is also regarded as the PRA synthetase, or the PRA is called the phosphoribosyl anthranilate. Now the tryptophan C help in the synthesis of PRA isomerase and INGP synthetase. The full form I will tell you later on. The tryptophan B and tryptophan A, these two genes express themselves in the form of tryptophan synthetase beta and alpha respectively. So 5 genes help in synthesis of 5 different enzymes. Okay, so they express in the form of enzymes. Now this is the biosynthesis of tryptophan, how the tryptophan biosynthesis occurs. Now tryptophan biosynthesis starts with the molecule chorismate. It reacts with the glutamate in the presence of the enzyme anthranilate synthase component 1, and it is converted into anthranilate. Now anthranilate reacts with the PRPP. The full form of PRPP is phosphoribosyl pyrophosphate. Okay, so it reacts with anthranilate in the presence of the second component, that is anthranilate synthase component 2, and converted into PRA or phosphoribosyl anthranilate. Actually these two enzymes, the component 1 and component 2, together form an enzyme complex, and that catalyzes these two biochemical reactions. Now the PRA is converted into CDRP, that is carboxyphenyl amino deoxyribose 5-phosphate, by the enzyme PRA isomerase and INGP synthetase. Okay, the CDRP is converted into INGP by the enzyme, by the same enzyme, and INGP is this indole 3-glycerol phosphate. Okay, so here also these two steps are catalyzed by the single enzyme. Now this indole 3-glycerol 5-phosphate 3-glycerol phosphate reacts with serine and converted into L-tryptophan by the enzyme tryptophan synthetase beta and tryptophan synthetase alpha. Okay, so this is the final product of this operon. So this is biosynthesis of tryptophan operon, and these are the components of tryptophan operon. Now coming to the regulatory part of tryptophan operon. How tryptophan operon is being regulated? So there are two modes of regulation in case of tryptophan operon. The first is the repressor operator interaction. Now the repressor sequence undergo transcription and translation to form the repressor protein, but this repressor is not the active repressor, and it is called as the aporepressor. It requires the presence of tryptophan itself, which acts as a co-repressor to form the active repressor. So here this aporepressor binds with the tryptophan, which is the co-repressor of this operon, and forms the active repressor. It binds with the operator and it will inhibit the forward movement of this RNA polymerase. As a result, the transcription of these 5 structural genes do not occur, and this operon becomes switched off in the presence of the tryptophan. While when tryptophan is not present in the cell, that means co-repressor is absent, this aporepressor cannot be converted into active repressor, it cannot bind with the operator. As a result, the RNA polymerase will move forward, and transcription of this 5 structural gene will take place, and it will form 5 different proteins that help in the biosynthesis of tryptophan. Okay, so in the presence and absence of tryptophan, the repressor operator interaction help to regulate the tryptophan operon. Now coming to the second mode of regulation, that is the attenuation, and this attenuation is very much important from the examination point of view. Now let us consider this thing. Proportion of full length transcript is inversely proportional to the amount of tryptophan. In the previous mode we have discussed about that the tryptophan is present or absent. Now the tryptophan can be moderately present, or the cell is starving for tryptophan. Okay, so moderate to very less amount of tryptophan can be present in the cell, and that amount is inversely proportional to the proportion of full length transcript. Okay, so this amount of tryptophan will regulate how much will be the length of the mRNA transcribed by RNA polymerase. Okay, how it will occur. Now the upstream sequence of tryptophan E is having this leader sequence, and this leader sequence is transcribed first before this structural gene. And in case of eukaryotes, the transcription and translation are coupled processes, and both occur simultaneously. Here within this leader sequence, it is having its own initiator codon, its stop codon, and between this it is having 2 independent codon for the tryptophan. It is also having 4 regions, 1, 2, 3, 4, which are complementary to each other. 1 is complementary to 2, 2 is complementary to 3, and 3 is complementary to 4. Okay, now if they are allowed, they can form secondary hairpin structure or stem loop structure. That means one can form hairpin loop or stem loop structure with 2, 2 can form a stem loop structure with 3, 3 can form stem loop structure with the 4, and these stem loop structure will provide different signal, okay, for the RNA polymerase whether to transcribe these 5 structural genes or not. Okay, this is called the attenuation. How this will occur? Now if the A bind with this 2 region and form a stem loop, it will give a pause signal. Okay, the transcription and translation are simultaneous process, and it will occur in a coupled way. Now when the transcription of this structural gene E, suppose E is going on, this portion is already being synthesized. Now whether to transcribe this whole sequence or not, that will be told by the secondary stem loop structure of these different regions. Now when 1 and 2 will couple with each other and form this secondary structure, it will synchronize the transcription process, the formation of this, with the translation process. That means binding of the ribosome. For that purpose, this 1 and 2 binding or secondary structure provides the pause signal, and it will pause the timing between the transcription and translation process so that they both occur simultaneously. Okay, so when the tryptophan is available to the cell, when tryptophan is very much high quantity, is present in high quantity in the cell, these codons, okay, these codons will be coded by 2 tRNAs, okay, in the presence of the ribosome, and that tRNA will have the tryptophan because tryptophan is present in the cell. That means the tryptophan tRNA is present in high concentration. In that situation, the ribosome will move from this to this side, that means the second side, okay, or second region. As a result, the 3 and 4 is now free to pair with each other, but as ribosome is present in the second region, it cannot pair with the first one, or second cannot pair with the third one, so third and fourth will form the secondary structure. As a result, it will give a attenuation signal, okay, because already tryptophan is present, it can form the tryptophan tRNA. That's why the ribosome is present in the first move from the first region. 3 and 4 form the stem loop structure. It provides attenuation signal, and the transcription of these structural genes is inhibited. Okay. Now, if suppose the tryptophan tRNA is very less in the cell, so the ribosome will remain halted in this portion, that means the first region. So the first region cannot form stem loop structure with the second region, okay, and as a result the second and 3 will pair with each other, and this pairing between the second and third will provide the anti-termination signal. That means it will tell the RNA polymerase not to terminate and move on and transcribe all these structural genes. Okay, so these 3 secondary structure or stem loop structure provide 3 different message. 1 and 2, pause, it will synchronize the transcription of this mRNA with the translation. 2 and 3, stem loop, will provide anti-termination, that it will say the mRNA, the transcription of the mRNA to proceed, and 3 and 4 pairing will provide the attenuation signal. That means the transcription process will be attenuated. Okay, so this is all about the tryptophan operon and the regulation of the tryptophan operon. So in the next video we will talk about the regulation of eukaryotic gene expression. Thank you for watching.